Good morning, everyone. Quick word from the organizing committee. First of all, welcome to today's session. Please try and help me with making sure that your mics are muted at all times. We will be having 3 videos that will be shared, and then there'll be a Q&A, quick questions at the end of each video, but there will be an extensive Q&A at the end. So if you have a burning question, please raise your hand and we'll get you on there. But if your question can wait until the end of the session, then that's also fine. There are 2 ways in which we can do that. You can either send your questions in the chat box, or you can wait for the session and unmute your mic and have an interaction with the facilitator. I will now hand over to the team that will be handling today's session to kick us off.
Thank you, Molati. Can you hear me, Molati?
Yes, you're on. Thank you.
Welcome to everyone to this workshop session from Metabolomics South Africa. To really thank you for joining to these workshops. There are going to be many series of workshops, trainings, and I will not take long. I will pass on to Dr. Sitore, who will introduce our first introductory workshop on ATMS. She will introduce our speaker, Dr. Edwin Madara from University of Venda. And I hope you will enjoy the sessions. As Molati said, for the questions, please type in the chat box and the moderator will take note of the questions, and there will be Q&A sessions at the end. Thank you. Dr. Sitore, the floor is yours.
Thank you, Dr. Tubismani. Sorry, I'm just trying to turn my video on. Okay, good morning. Thank you, Dr. Tubismani, for the very warm welcome. And a special thank you to ACGT and Diplomics, as well as Metabolomics South Africa, for organizing and planning today's training workshop. As we all know, today's training workshop will focus on LCMS, specifically sample preparation, sample metabolite extraction, and data analysis from samples that are analyzed using LCMS. So the session will be facilitated by Dr. Edwin Madala, who is currently at the University of Venda. And the session will be focused mainly on nutraceuticals, which is Dr. Madala's primary research interest. So the session will, like Molati said, it will be made predominantly of a pre-recorded video, but Dr. Madala is available to go live for any clarifications or to answer any burning questions. Now, just a short introduction. All of you received the email and all of you have seen his bio, but just a short introduction. Dr. Edwin Madala started his scientific journey, interestingly, at the University of Venda, which is where he is now. So he obtained his bachelor's in biochemistry and biological sciences. Then he later moved on to the University of Johannesburg, where he obtained his master's and his PhD. Since then, Dr. Madala has been primarily working on phytochemistry with a special interest in nutraceuticals, which we will see today. Also, Dr. Madala has won numerous awards. In 2019, he actually received the awards for most promising emerging researcher, and he has published over 70 peer-reviewed articles in reputable journals, and he's also published a couple of books. So without going too much into the details of who he is and his research, I think we should get to the videos because it's actually very interesting. So I think, Molati, if we can watch the first video. So ladies and gentlemen, after the first video, if you do have burning questions, we will take 2 or 3 questions. You can then type the rest of the questions in the chat. They'll be read out. And then as the videos go along, we'll stop for questions and any comments. Thanks, Molati.
The first step in metabolomics studies is sample preparation. Today we are going to use a plant called Moringa oleifera. Moringa oleifera is a plant that comes from India that is being used for nutraceutical purposes. We are going to use leaves, so we're going to homogenize our leaves. So I'm just going to take some leaves, put them in this blender, and then squeeze them in, and this blender will then pulverize the leaves into powder, and this is the powder that we are going to extract our metabolites from. So I'm just going to remove the stem part so that we are left with a lot of leaves in there. So it's very important that when you do this step, you keep it uniform so that the experiment will be reproducible. So this is a very, very critical step. So now you start blending like you normally blend in the kitchen. Just less than a minute. You can check whether if the leaves have been pulverized. Again, we try to blend it further. Pour it now to in a place where you can weigh it from. There's still some parts, the stem part in here, but this is enough powder for today's experiment. Just again try to remove the stem particles that are here. What we do in our laboratory, we have established a protocol where we extract our leaves using 80% methanol, because we found it to extract a lot of metabolites that we are targeting in this plant. So the first thing to do is to weigh now your 2 grams into 2 different tubes. We're just going to do 2 extractions for demonstration. So basically when you do this type of work, you do it in multiple tubes so that you can have a little bit of reproducibility. So I'm just going to zero my scale. So from zeroing the scale, you weigh in the 2 grams. You're gonna weigh the 2 grams. You must make sure that you are exactly at 2 grams. So this is good enough. So I'm gonna transfer this to a tube now. Do the same for the second tube. Again, you must make sure you are exactly at 2 grams. To the next tube. And this would be your leftovers. I'm just going to move it aside. We're done with these leaves. I'm going to move it aside. And now, the next step, as I've already mentioned, is to mix now your leaves together with your 80% methanol. We have found that 80% methanol works very well for extracting polyphenolic compounds. We have tried 50, 60, 70, and 80 to 100% of methanol, and we have indeed found that 80 is perfect. So I'm going to put 20 mils of 80% methanol in each and every tube containing my leaf sample. I'm going to put it in here, and then you close in now your tube. That's to be tube number 1. You mix a little bit so that you create a homogeneate. Then you do the same for tube number 2. Again, I'm measuring 20 mils. And then the next step now, because we want to aid the extraction, meaning that the removal of metabolites from the leaves to the solvent, we then put it in our shaker, which will allow that to take place. So I'm just going to put it here. There are other samples from other experiments that we're currently conducting. And then from there, what you then do, you allow this to shake for whatever time that you deem to be sufficient for your metabolite extraction to take place. So this is the next important step. Sometimes this step, you do it for an hour, 3 hours, or even 24 hours, depending on how much extraction you want to aid. So our extraction has been taking place for 24 hours now. So we are going to take them off from the shaker, and then now we're going to take them to the centrifuge. So I'm going to stop my shaker. So I'm going to remove my samples, the 2 samples that we prepared 24 hours ago, and because there's another experiment that is taking place, I will leave this to continue to shake. Then the 2 samples, we then take them to a centrifuge. So you put them in a centrifuge, then the centrifuge will separate between the material, plant debris, and the solvent, so that we can get solvent on the top. Now you close your centrifuge. You set the speed of the centrifuge, which is going to be 5,000 RPM, and we're going to do this for at least 10 minutes. And then start. After 10 minutes of centrifuging, the instrument will warn you that it's end of the run. Then when you do, you open the centrifuge, take off your samples. Make sure that when you take your samples, you don't disturb them much, because you don't want your plant leaves to mix with the solvent again. You want them to be 2 phases. Then you will see, as I take the first sample out, that indeed your cell debris are at the bottom, and the supernatant is your solvent, which is your methanol in this case. So you take the other one, make sure that you don't disturb it. The same thing has happened here. The cell debris at the bottom, and then you have your supernatant, which is your solvent, and you take it back to the workstation. Now we are coming to the most important part of our experiment, which is now to filter your samples for analysis on the LCMS. Then you have 2 vials here, my 2 vials, my vials, and then I put the inserts inside my vials in order to minimize the volumes that we're going to use on the LCMS. Normally LCMS inject microliters, so we have a lot, so we just have to minimize what we put in our vials. Then we put the inserts in, and then you open up your syringes, the first syringe, and this is very important that you use each and every new item all the time, so that you don't get sample mixability. So each sample must be treated independently. So you suck up with your syringe at least 1 ml, even though your insert takes 200 microliter, you take more than enough. At least in this time, in this experiment, we have enough sample. So you put now your 0.22 micron filter in front of your syringe, so that if there's any debris in your solution, it can be eliminated by the filter. So you filter through your extract into the vial. You allow it to drop, do it drop-wise until you fill in your vial. And then you can cap your vial and keep your sample aside, and then you move to the next sample. So you do the same. Make sure that you use different syringe again. You suck up the liquid, so you have done in the previous one. Again, you put in a filter, a new filter all the time, and these are not reusable, as I've already mentioned. Make sure that you don't do this fast, because you can damage the integrity of the membrane. You allow it to drop into the vial until it is full. When it's full, you can close your vial. Now you have your 2 vials. You can just tap them to remove any form of air bubbles, and then your samples are ready for LCMS.
Are there any burning questions at this point, or should we move on to the next video? Please raise your hand if you have any burning questions. Let me. Okay, let's proceed on to the next video then.
Actually, there is a question. I'll read it out. It's from Marlies. So Edwin Marlies wants to know, what type of syringe filter do you use? Also, is methanol the only solvent that can be used for such extraction?
Thank you, Lungi. Thank you for the question. Yes, there are different types of filters. We use the nylon filters because we found those to be good for the type of samples that we work on. However, I've also seen other people using different types of filters, but it also depends, as I've said, the solvent that you're using, also the type of sample that you're using, because some of the filters, depending on the integrity of the membrane itself, they can wash off, depending on the solvent, to your sample extract, and then you end up seeing them as part of your analytes. So these filters that we currently use, we have been using them for years now, and we found them to be useful for plant metabolites. You see, the difference, as I've already said, is a sample type, because if you're working on mammalian stuff, you guys, you know, you work with very tiny amount of volumes. So sometimes you might see that the metabolites that are derived, or signals that are derived from your filters can interfere with your metabolites. So in terms of solvents, no, we don't only use methanol. We use different solvents. We can use ethanol, we can use chloroform, we can use ethyl acetate, but it's just for the sake of analysis on the LCMS, we mainly reconstitute our samples on methanol, because we found it to be appropriate with our instrument, because sometimes if you use some corrosive solvents like chloroform, we really don't advise that people use chloroform on the LCMS. So yeah, so basically ethanol is the superior one, but you can use almost all solvents. But just be careful on what you use and what type of tubing does your LCMS have, because some solvents can be of danger to that. Okay, I see a lot of questions here. I see someone ask about, okay, Dylan has already answered that we use 0.22 micron filters. You can use, 0.22 is fine for LCMS. We have started some time ago, when we were still working on HPLC, we used to use 0.45 micrometer size filters, but we found that also results in some of debris going through, and that could be detrimental to your instrument. So 0.22 micron is good. And someone asked, is it a deteriorated methanol? No, it's a normal methanol that we use, unless if you are using deteriorated standards. We also use normal methanol for that, but the standards will be deteriorated in order for you to differentiate with a normal analyte. But as for the solvent, this is a normal methanol. And
a quick question. Sorry?
No, I saw another question. If someone asks us, can you use the same extraction method for bacteria? My answer is that there's no golden method for extraction. Like I already said, we use various types of extraction methods. So like I said, some students here in my lab use 40% ethanol, some use 80%. It's just for plants and the type of metabolites that we are targeting, we use 80%. So as for the bacteria, I know my friend at UJ, Dr. Tigizimana, they work on bacteria. Dr. Mthongo, they also work on bacteria. Maybe they can assist what solvent they use there and whether if that it gives them better results. But as for plants, we use 80% methanol, but we can change the percentage of methanol. Okay.
Edwin, I think I can allow two more questions. We move to the next video, and then we can always come back to the questions right at the end. Let's see. So I think there was an interesting question, Edwin. I'm not sure if you answered it because I did go black there for a little bit, but let's continue, Mulati, and then after that, we'll get back to the questions.
So we have arrived in our LC. So I open where there's a sample compartment. These are different trays. It can take 3 different trays. I'm gonna put my samples in the sample compartment, number 1 and number 2. Close in my samples into the sample compartment, and then I move to the control computer where I'm gonna start opening my method, and the method will be based on the folder depending on the project. Then open the method depending on the project. I'll go to the appropriate folder. I'm gonna open the method. Once the method is open, I'll then check the parameters. Once I'm satisfied that the parameters are okay, the method is the one that we're going to use, then I'll start the run using a single run tab. Then now I will direct where the data should go. So I'm gonna choose a folder where the data should go. Then I will call it Moringa 1. And I will tell the instrument where is the sample. Then is it vial number 1 of tray number 1. It must inject 3 microliter. And once I'm done, I can now start the run. So now the instrument is just getting itself ready. So once it's ready, the injection will be started. So as you see now, it's starting a new run. Everything became blank. Now a new run has started. As you can see with the trays here, the new run has started. The mass spec is starting to acquire the ions that are coming from that particular sample. And then you will see now when a first signal comes in, and then that's when I'm satisfied that indeed everything has went very well. As you can see here, the method that we're currently using runs for 53 minutes. Then what will then happen is that the instrument will acquire data for 53 minutes. And once 53 minutes is done, the data will be ready for analysis. Then we shift the data from this computer to the data analysis computer where we can look at the success of the run. This is the computer that we use to control our instrument. And then this part of the instrument is the LC part, which is the liquid chromatography part, and it's equipped with a PDA detector. From the PDA detector, the samples that are eluted from the LCMS part are then transferred through this small tube to a mass spectrometer. So this whole box is a mass spectrometer. We have a very fascinating instrument here in front of us. This is a quadruple time of flight mass spec from Shimanzu. It's called the 9030 model. What it does is that it can profile metabolites. It can also quantify metabolites. And the speed of this instrument is unprecedented. It's one of its kind. It's very fast, hence it can do both profiling and quantification at the same time. So in order for us to get our samples ionized efficiently, we then use we use the nitrogen generator, which is connected to the LCMS. Previously, they used to use cylinders, but these days, these instruments come with nitrogen generator. The nitrogen, which is generated by this instrument, is used for ionization and drying of samples. We are based in an area which is prone of electrical cuts. So our instrument is connected to a UPS. So this UPS can sustain this whole instrument, including the data computers and everything, including the generators, for at least 3 hours before the electricity can come back. So between the power cuts, the UPS keeps our instrument on.
So once the data is acquired, you need to now then convert it into an open source format that you can upload to different online platforms to analyze that data. So for this Shimazu machine, we have a system called Lab Solutions where we can convert that data. So once you open Lab Solutions, you can then go to your post-run analysis where you can then convert your specific data to an MZML file, which is open source and can be read with various online platforms. Then it will start that conversion over there. So now once the data has been converted to the MZML, you can find it in the folder that you directed it into. And then to get the particular data set to go online, you can use an FTP server. The one that we use is called WinSCP. So in WinSCP, you then log in using your GNPS details. Then once you copy your data into this FTP server, it will be uploaded online where we are going to do the majority of the data analysis. Alright, so one sample can produce about 100,000 kilobytes of data. So if you have multiple data sets, it will be better to upload it through an FTP server so that it's much faster and it's much more reliable to do it that way. The next step after uploading your data in the FTP server is now to come into GNPS, which is Global Natural Products Social Molecular Networking. It's a freely accessible online platform that you can do molecular networking and many other computational tools that you can use to analyze your data. So today we'll be doing classical molecular networking, which then allows us to view the chemical composition of our different plants that we are running in this lab. So once you open up your molecular networking workflow, first you will log in, then you will open up your molecular networking workflow, then you will give your job a name. And then once you've done that, you can select your input file. That input file, we uploaded it earlier on using the FTP server, so it will then automatically come online. Then you will select your data file that you have uploaded and then finish your selection. That data file will now be uploaded. Now there are various parameters that you can set so that you can get, you can control how your molecular network is generated. First, there's precursor ion mass tolerance. What is nice about GNPS is that all these parameters are predefined by you, are defined for you so that you can look what best suits your experiment. So we have been working on this for a while, and we know for this data set, a mass tolerance of 0.05 Daltons is much suitable. There's a lot of advanced network options that are listed for you there, and then you can also look what each thing will affect, how each thing will affect your data set. Once you have set all those parameters, you can then submit your job, and then it will email you. Once the job is done, then you can come back and visualize your job. Then you hit submit. It will automatically start computing your job right there, and this can take from minutes to hours to days depending on how many data sets you have uploaded onto this system. So once this is done running, it will notify you, and then you will go to your job status page where you can download and visualize your networks. On the status page, it will show you that your job is done, And then here you can then view all library hits. So what this molecular networking does, it will also compute library hits to matched compounds that are out there. So you can view that, but for illustration today, we'll just be going through the classical molecular networking, which I will then download a direct cytoscape preview. Then your whole network that is computed there will be ready and accessible for download. Then you download it, and then you can open it, which will then be ready to view on cytoscape. Classical molecular networking uses a spectral alignment algorithm, specifically called MS cluster algorithm, which aligns all the spectra in your dataset to each other and also matches those spectra to known compounds that are out there. So each node, the circles that you see here, will represent different consensus spectra, and then they are connected with edges which show the relationship between these compounds. So this will be grouped into a molecular family depending on their structural relatedness. This is based on the fact that when compounds that are similar in structure fragment, they yield similar fragmentation patterns. So this algorithm that is built into GNPS can then identify that this particular compound, for example, the rutin that we see here and camphoryl rutinoside that we see here, are structurally related based on their MS to the 2 spectra that we uploaded. This then helps us to identify and visualize the chemical space that is within our plant. It then allows us to view this plant in a more global way and get as much information from the data that we extracted. In a nutshell, what molecular networking does for us is that it is able to compute for us which compounds are structurally related, assisting us in metabolite annotation and viewing the full chemical space that is within our plant.
Thanks, Malati, for the videos. So we have about 5 questions that were not answered. We'll go back to them. But before those questions, may I ask Lillian to maybe unmute and ask her question? Lillian?
Hi, can you hear me?
Yes, we can.
Yes. Good morning, everyone. So my question was, I am doing a study on papaya peel extracts, and from my research, I found that the best extraction solvent to use would be acetone, 90% acetone, right? And that's what I use for my extraction. But then I had to identify the active compounds within the extract. And to do that, if I'm not mistaken, you're supposed to use the same extracting solvent you use for your other experiments. But then the machine that was being used, well, I spoke to the lady in charge of the machine, and she said we can't use 90% acetone for the extraction to do the LCMS because it would damage the columns of the LCMS machine. So the option we used was to use methanol. So my question was, the active ingredients that I identified within the extract, is that going to
be used acetone, but for your metabolite stuff you use methanol?
Yes. So is there going to be like a change in the compounds?
Can I answer?
Yes, Dr.
Yes.
Madala, please. Edwin, just unmute yourself, please.
Okay, I'm trying. Okay, can you hear me now?
Yes.
Yeah, I was saying to Lillian, she's very, very right. She extracted with acetone, but unfortunately we can't run acetone on the LCMS because yes, indeed, the technician told you right. It damages the column and also the tubings of the LC. So normally what we do is that even though you have extracted with acetone, you dry it out and reconstitute your metabolites or your extract into methanol. Yes, indeed, you're going to lose some of the compounds, you're going to lose some of the amount in terms of the quantity. So some metabolites prefer to dissolve in acetone. So just to give you a perspective how much of the change can happen due to the solvent change, it also happened with the same solvent. If you use the same solvent at different temperatures, if you use the same solvent, for say maybe methanol, from different vendors, you buy one from Sigma, you will buy one from another company, you are still going to see differences amongst those extracts. So imagine now changing from acetone to methanol. There's going to be extremely huge changes. So this is the compromise that we have to live with, especially when we work with LCMS. It's NMR where you can work with almost all type of solvents, which as long as they're deteriorated. But with LCMS, yeah, we try to preserve our instrument as much as we can, and that's why we prefer to use methanol. So what I normally do, sorry Aurelia for keeping my answers too long, but what I normally do is that we extract with different solvents, say acetone, ethanol, methanol, could be chloroform, could be hexane, and we dry them out and we reconstitute them to the same solvent, meaning that we assume that whatever that we lose on one, we lose on the other. But however, you must also remember that's not how, you didn't start with the same thing. So with the hexane, you will extract a lot of non-polar. With acetone, you will go for mid-polar, mid-polar. So yes, you are going to see changes, unfortunately.
All right.
Thank you, Dr. Madala. I will take only 2 more questions and then I'll read the rest out. For those who still have really burning questions, please go to the Metabolomics South Africa website. I think there will be a forum created on there where you can ask your questions and Dr. Madala will assist and answer your questions accordingly. So Leah, I'm sorry if I'm mispronouncing your name, but you said you had a question. Please go ahead and ask your question. And then that will be followed by Johnson.
Okay, good morning. Can you hear me?
Yes.
All right, so I am not sure if my question should be asked now or I should have waited until the end. I just wanted to confirm because I'm working on bacteria, on bacterial samples. I don't know, on your platform, do you work with bacterial samples or you only work with plant samples? Because right now I'm at the point where I'm looking for where to go and do this analysis. I will be doing proteomics and metabolomics. I asked the question with the misconception, thinking that this workshop is coming from University of Pretoria area, which was a mistake. So I don't know if in your lab you work on bacteria, and if also you also do proteomics, so I can take the discussion further, acting, you know. Yeah.
Yes, Ilya.
Secondly, I want to ask, what of the use of Galaxy for the data analysis? As in what
the use of what?
Galaxy platform instead of the GNPS.
Okay.
Yes, so yeah, these are my 2 questions.
Okay.
Thank you.
Thank you. Maybe for the data analysis, I must say the way why we are using that platform is because our colleague, Dr. Tugizimana, who's mainly focused on the data analysis side of things, he has active collaborators with the developers of those molecular networking, which of course, the reason why we chose that platform is because if we happen to correspond any challenge, at least we have direct contact with the developers so that they can assist us with any question that we have. Anza here and the other students at University of Johannesburg, they are working on that platform, and they have direct contact, and actually they are co-supervised by people who have written those algorithms. That's why we are there. So now coming back to bacteria, here at the University of Venda, yes, indeed, we have a very strong department of microbiology. I want to advertise it and even say it's stronger than any you can imagine. Of course, we can support bacterial work there in that department. And of course, LCMS can analyze any chemical regardless of the source. Even if it's coming from the bacteria, come from a human being, come from a plant, come from a frog, come from a fish. As long as it's a chemical, LCMS will pick it up, which therefore means as long as you can culture your bacteria and be able to extract the metabolites from there, we are able to run them on our LCMS and be able to tell you what is going on physiologically. So yeah, I'll make a note about what we can do later on after we have answered all the questions. Dr. Stolle?
Thanks, Dr. Madala. So I'm going to combine the next three questions because they're a little bit similar. So Elise wanted to know if the MS analysis was done in scan mode, and if does GNP annotate using spectral libraries, and do you get a spectral match in terms of percentage? And then Johnson's follow-up question was then, how can one improve the use of nutraceutical quantification on LCMS and GNPS? And then also, Zoteti wants to know, is the data pre-treatment step still necessary when using GNPS?
Yes.
Okay, I think you can answer those two, yeah.
Okay, unmute your mic. I'm gonna give Anza a chance to just run through the.
Hello?
Are you unmuted?
Yeah, I think I am.
Continue with the GNPS.
So I'll start with the first question to the best of my understanding. Is the data pre-treatment step still necessary when using GNPS? So it depends on what you are doing. So with classical molecular networking, what we do is just convert to the MZML files in Centroid. So it uses MS2 data. But if you're doing something like feature-based molecular networking that takes into account things like your chromatographic information, you might need to use pre-processing softwares like MZMine, I forgot the other MSDAL, to perform some sort of pre-treatment before uploading your data set. So it depends on what you want to do. And then if I can find the other question.
So Dr. Mandela, how accurate, or Anza, how accurate is the GNP system? Because somebody did ask that.
Okay, let me come in there. Basically, and to first answer the question on scan mode, yes, we do a scan mode. But how accurate? The accuracy of identification by that platform is dependent on the type of the instrument you use, on the accuracy of the instrument. That, remember, molecular network is not only based on the fragmentation spectra only, it's also based on the accurate mass that you impute in. Remember, when you do MS, you must have a precursor, you must have product ions. So I work with isomers here at the University of Venda. So isomers, you know, they are they have the same mass, but sometimes when you fragment them, they will give you differences in fragmentation. So before we run anything on the molecular network, we must first train the instrument. We train it by using known compounds so that we can see whether it is indeed predicting to the best level that we can. That means you can even, for instance, put a known compound in your extract as an internal standard, so that when this molecular network is predicting the identity of the molecule, we can therefore even look at the internal standard if indeed it has identified it precisely. So these database spectral alignment libraries that we are using, they are based on accurate mass and also fragmentation. So before we even go there to the molecular network, we have to train our instrument. We have to get the proper collision energies. We have to know what type of, how long should our run be, whether there's a good separation between isomers so that we can differentiate them. So yeah, it's a lot of work in terms of optimization until you get to the level where you can say now you are going for data analysis.
Thank you, Dr. Madala. So in the interest of time, I just want you to give like an umbrella response to some of the questions we missed on the most technical aspects of sample preparation. But before that, can you just highlight, does the software annotate or does it also quantify? And if somebody's interested, being a protein person, would this also work for them? Very short and sweet, Dr. Madala, please answer those two so that you can answer the other questions.
Thank you very much. I'm not that short, but anyway, it can do proteomics. We can do proteomics, but we are still at the very early stages of optimizing our instrument to also handle proteins. We have done a master's project on it, working on proteins. It worked very fine, but we are still at very early stages. Secondly, this is one of its kind. This instrument can do quantification at the level of a triple quadruple. So if you are familiar with Shimanzu instruments, they have different types of different types of triple quadruple. This one can operate as a triple quadruple, and meaning that it can do quantification at the level of an 80-40 instrument, which is amazing because almost all our chemistry students here, they do quantification. They get very nice calibration curve, and it's so sensitive that we can even go to a single-digit ppb level. For a QTOF, that is amazing. So yeah, we can do quantification. And then in terms of annotation, yes, we just got an interesting software now, which is called an Insight, where it can, of course, calculate the elemental composition of each and every signal it picks up on a chromatogram, give us an elemental composition. We can use that elemental composition to search against online databases, and then to a certain extent, some metabolites we can annotate automatically. Some we have to do it manually because we have to work with the energies, collision energies, in order to get the structural features that will, of course, encourage us to go with the identification that we choose.
Thank you, Dr. Madala. Very comprehensive response. So the last question, for those starting out in this type of work in metabolomics, also for those who are still interested in working with Moringa, can you please just comment, just confirm that when doing the extraction, 80% methanol would be methanol and water? Is that what 80% methanol is? But also, your samples were in plastic tubes. Does the plastic affect the extraction procedure? And how long can you keep the powder after homogenizing it? So how long can you keep it for freshness? Lastly, when drying your leaves, why do you dry instead of freeze dry? Which would be the best option? So I think once you've answered those, yeah.
Thank you. Let's start with the drying. We do all kinds of drying. We have the first dry in our laboratory, as you have seen next to the centrifuge. Immediately is the first dryer. We also do first drying depending on the type of sample. Secondly, going back now with regard to Moringa, yes, we are doing a lot of Moringa work. We can even brag and say we were the first group when I was still at the University of Johannesburg to actually comprehensively publish the Moringa metabolome. In terms of all the flavonoids that are there, we are able to spot them without even an MS instrument. We dream them, we live them, we know all of those. So again, plastics, yes, stay away from plastics. Unfortunately, we don't have glass centrifuge tubes, but as much as we can, we need to stay away from plastics, especially if you're working with sensitive analytes, and depending on the solvent, as I already mentioned, the plastic can wash out and get into your analytes and it can interfere with your signals. So what was another question again? Ethanol. For Moringa, if you want to get almost everything, I will tell you 80% methanol is the best way to go.
Okay, thank you, Dr. Madala. I do want to say we are done, but I do see Johnson's question is burning because he has now typed it the second time. So final comments, Dr. Madala, can any type of LCMS be used? Can HPLC be used for metabolomics? And how do you use standards to quantify the concentration of neutrophils in the sample?
Thank you. not every LCMS can be used for metabolomics. You know, it depends on what you want to do in metabolomics. We say rather use high resolution mass spectrometer. Your quadruple time of flight, the likes of your MAL-DTOF can also be used. You can also use your the likes of Orbitraps. But you see, there's a problem, especially I would say in South Africa, where people want to do LCMS type of work, and metabolomics work, and they go and buy an instrument, and then they just buy whatever that is cheap. And then when they try to do this type of work, they can't get to the level where we are, basically because they didn't do their homework properly when they buy instruments or when they analyze their samples on any instrument. So no, there are instruments that have certain accuracy level that they can be able to be used for this type of work. And then we bought this instrument basically looking at how accurate it is. And then indeed, in terms of quantification, it has a mode which is called the multiple reaction monitoring mode, where of course you inject your standard, you study its fragmentation, you get the precursor ion, product ion, and with that combination, achieved is actually from a triple quadruple, but this instrument is amazing. I'm not on the payroll of Shimanzu, but I'm just putting out there, it's an amazing instrument, this one. Lungi, if I can say something before we close.
Very quickly.
Is that our University of Venda, we have this instrument. For the first time, maybe you will hear this, you never hear it anywhere else. Our instrument, we are open for public. You can come and use our instrument as a means of collaboration. You can cover your own cost to come here. You can cover your own cost for your samples. We will give the instrument for free of charge, as long as you can acknowledge us through authorship of your manuscript, so that we can generate revenue to keep our instrument going. We don't charge a single cent for analysis here, but we, of course, look at the project at first and then see whether if we'll be able to help you. We are not willing to go further than analyzing your instrument in terms of data analysis. We can maybe send you to Gizmana at UJ, where you can learn data analysis. But in terms of the hardware, we are all available for that. And we are again buying another instrument, which is going to be only for quantification. So we want to create a center of mass spectrometry at University of Venda. This is our dream, and we hope everybody can support us, but we are moving there. Thank you, Lungi.
Thank you so much, Dr. Madala. For those questions that were not answered, you will be given an opportunity to send them via email, and Dr. Madala will respond to them. I am sorry that we couldn't take all the questions, but in the interest of time, we do have to cut it short. But once again, thank you so much, Dr. Madala, for the very interesting introductory session. And I think perhaps the organizers should organize a follow-up session, right, where we go into much more detail, because there are quite a lot of questions. But for now, I will hand it over then to Dr. Aurelia Williams, who will then do the closing remarks, and she will add on anything else that needs to be added. Thank you.
Dr. Sitole. Can you guys able to hear me?
Yeah, we can hear you.
So I think, is there any pressing question? I think we can maybe take one or two more. Is there any question we haven't covered? Perhaps the individual can just unmute if there's somebody with a pressing
Abidemi,
question.
I see Abidemi's hand is up, so maybe he can unmute himself and ask his question.
Yeah, thank you very much. And I want to appreciate the organizers of this workshop for a very useful and insightful presentation. I just want to make a request. Are we going to get a transcript of this session? Because I see that this session is being recorded. Will it be possible for us to get a transcript of it so that we can actually go back and then listen to some of the discussions that has gone on in this workshop today? Thank you.
So we will make available, I think, the recording as well as the individual videos. Mulati, we will post that as well. I think before I actually close, I'll give him an opportunity just to direct us and let us know where exactly we can find everything.
Yes, I'll give that information towards the end.
Okay.
Aurelia, maybe take Mpepu's question as well. It's typed in the chat box.
Dr. Madala, so I'll take this last one. Just before I think, how does sample collection and storing, how does that have an effect? Does it have an effect on some metabolites? Is an approved protocol for sample collection and storing sample?
Thank you, Dr. Aurelia Williams. It's a very good question. We work with different type of samples. Of course, for instance, the type of work we do, we work on geometrical isomerization of molecules. When we collect our plants, we make sure that we keep them in dark. Even when we dry our samples, we keep them in dark, depending on the type of experiments we are going to do or establish. So one has to be very careful, especially if you are working on, for instance, biological influences. You have to quench the metabolite using liquid nitrogen. That means those will be difficult to go to the field to collect, because you're gonna have to go there with a nitrogen canister so that you can keep them cold, so that you can quench all metabolic processes. At UJ, we use our plants. So some plants, of course, we keep them at -80 degrees Celsius. Some we keep them in plastics, some we keep them open. So it depends on which type of sample you are working on. So especially environmental samples, we don't really preserve them, but we have noted that, of course, handling them, depending on how you handle them, there's going to be a lot of changes that is associated with handling of plants. So you have to treat each and every sample differently. That's all I can say for now.
So just on that note as well, I think everybody should just keep in mind that with Metabolomics, I specifically focus on human-based samples. So my focus is HIV or infectious diseases and cancer. So I think what we should keep in mind is that you cannot just delve into a particular experiment. There's a lot of optimization that needs to happen. So you can't just assume that, oh, I can store my samples for so long, or I can store it in this way. So with each project that do optimization, and you need to check. In our case, for example, we cannot just assume with the different cell lines that, chloroform, methanol, water, that combination will work. We do different combinations of solvents, and we test it for each cell line that we work with. So I think the message is that you definitely need to do optimizations for your particular experiments. And if you really struggle with finding differences, then I think your second resource is then find a backup, literature to support why you're doing X, Y, and Z. Okay, so with that, I'd like to say thank you to Dr. Madala for availing himself, as well as, Diplomics for participating and assisting us in this. We look forward to building this future collaboration and the partnerships. And then, of course, University of Venda for actually, hosting us and allowing us to do this. Then, of course, I'd also like to say thank you to the ACGT for helping us, especially with the administrative half of things, particularly Mulati, for getting all of the information out to the, the different stakeholders, to all the delegates, and then making sure that all the technology is in place for us to actually do this. I'd like to say thank you to Metabolomics South Africa, for Dal and team, all the subcommittees for their various inputs, assistance, insights that they give, and even the relevant suggestions. And then, of course, lastly, I'd like to say a warm thank you to all of you delegates for actually participating, because this is actually for you. And, part of the introductory work, a lot of us might assume that many individuals out there actually know or are aware how to do these or follow these workflows for metabolomics. But I think from this, we see that there's a definite need for even just the basic introductory stuff. So I'd like to say thank you to everybody. And then I'll hand over to Mulati just to indicate where we can find all the recordings and where all of this will then be placed. And perhaps maybe just Dr. Madala, Dr. Sitole, if you guys can maybe just put your email addresses in the chat. I'll hand over to Malati.
Okay, thank you, Aurelia. Thank you to all the participants for showing up today and making this a lively session. To get to this point, there was a lot of meetings, a lot of ideas, and a lot of time that was donated generously by the people in the subcommittees and in the committee of directors of MSA. And I would like to thank them all under what Aurelia has already said. I would also like to thank Dylan for finding some of the questions there in the chat. Your efforts were noted. But this session was recorded. The videos will be available. We'll put YouTube links onto the MSA website and the ACGT website, and I'm sure Diplomics will also share some of these. You should all have my email or my contact information if you need to get in touch with any of the speakers that were here today, or you would like to, because part of this is to build relationships moving forward. So if you feel you need to collaborate with Dr. Madala, for example, I can try and facilitate that for you. So all the information about this session, the recordings and the videos will be available specifically to people that registered this, but they will be available on those websites. So once again, Dr. Madala, Anza, thank you very much for a very productive session. And we hope to see you guys again in future sessions. Thank you.
Thank you very much, Mulati.
Mm-hmm. And with that, I would like, unless the chairman of the MSA would like to say something, Fidel. I guess not.
Mulati, can you hear me?
Yes. Okay. Sorry for putting you on the spot. I didn't prepare for that one.
Yes, you did indeed. But yes, thank you. Just to what Aurelia and yourself said, to thank everyone who attended, and mostly Edwin for your presentation. It's really insightful, and looking at the questions, there is indeed a need for a follow-up on the topic. There are quite a number of practical questions that still need to be answered. Thank you so much, Edwin. I'm sure we make a call on you again for more input, and please stay tuned. There will be more workshops on different topics, and they will be advertised via ACGT or MASA website. And Aurelia, thank you so much for organizing this and for your really dedication for workshop trainings and gathering the skills like Edwin to give these talks. I'm sure there will be more talks, not only on plants, but also for human-based metabolomics. And there is a lot that will be given. Stay tuned. And for data analysis and the other aspect of metabolomics workflow, it should be, we are planning for more trainings and at different levels, introductory and advanced training. Mulati, thank you so much for all the assistance and having this successful workshop. Thank you so much.
All right, everyone. See you next time. Bye-bye.
Bye.